Alright, let's do this. Let's go over the 1.1.4 crime scene evidence analysis notes, meaning here's some background information, some other essential stuff that you might want to know in order to understand why we actually have the ability to use some of these pieces of evidence as ways to solve the Ana Garcia crime. So let's get into it. We're going to just get right into the hardest one, which is actually blood. In order to understand why we're able to use blood as evidence in a crime scene to either eliminate possible suspects, to place somebody at the scene, to determine whose blood is actually at the crime scene, you have to actually know some of the genetics behind it. So let's just kind of get into that real quick. First and foremost, you are you because you got half of your genome or genetic information from your mom and the other half from your dad. The two of those coming together, combine, interact with each other, boom, you end up with you. Each of your characteristics that are genetically determined are actually called traits. You need to realize that traits aren't just this thing that are the physical characteristics of an individual, such as the fact that I have a black beard with gray hair right here, or the fact that I'm bald, I have brown eyes. None of those things, well, I mean, actually all of those things are in fact traits. However, you need to understand that it's also other things, such as your digestive system's ability to break down lactose, or the inability to, or having sickle cell, or any of these other things are all characteristics that are kind of behind the scenes working that are still determined by all of your genetic information. So if you break it down to an individual trait is determined by a sequence of DNA. That sequence of DNA that actually determines a trait is called a gene. Sometimes you can have a characteristic or a trait that is determined by just one gene, so one segment of DNA in one location can determine things like, if you think about Mendelian genetics, maybe the widow's peak that occurs at the top of somebody's head, those are just single gene traits. You also have those that are multiple gene traits, meaning there are several small sections or several sections of DNA. They don't have to be in succession, they can be in the same chromosome, they can be on different chromosomes, but in combination they actually come together to determine a trait. One of the most common ones that we know are multiple gene traits are eye color, and in this instance, blood actually ends up being a multiple gene trait. So if you look at the entire human population, you'll also see that the entire human population might have different versions of a gene, and that's evident normally in their physical characteristics. But through analysis, we've actually found that just because there are different characteristics doesn't necessarily mean that a different characteristic is a different gene. So when we break it down genetically speaking, we can see that if there are in fact different versions of a gene, we give that term for the different version an allele. So for example, if we're talking about blood, there are 3 different alleles for blood. There's one allele that determines the type A, one allele that determines type B, and an allele that determines type O. But you'll notice when we've been talking about blood, we've actually referenced that there is type A, B, O, and AB. But we're talking about versions of a gene or alleles. AB is just a combination of 2 alleles. So it's not its own version of a gene, it's just simply the combination of 2 versions. So something you also need to understand is you as an individual, for any given trait, you only have 2 alleles for a given trait. That's simply because you receive one from mom, one from dad, come together, you get 2. So there can be different combinations of alleles, and those different combinations of alleles that come together are called a genotype. That is the gene combinations. So for example, let's say your mom carries the allele for blood type A, and that gets passed down to you, and your dad carries the allele for blood type B, and that gets passed down to you. Well, those two combine to form a genotype of blood type A and B. Well, then the physical expression literally means that your red blood cells will be caused to have the expression or the actual physical appearance of AB blood type. So, referencing back that again, blood is an example of multiple alleles, and in the human population we have 3 alleles for blood type. And they're referenced as, right here, this is their abbreviations for the alleles. So you have the allele that is for blood type A, is I superscript A. You have blood type B, which is I superscript B, and blood type O, which is I superscript O. Now, the mode for which these blood types will rise about has to do with simply when one of these alleles, when one of these alleles, like blood type A, is given by mom, and the other allele, let's just say for instance it happens to be this one here, is given by dad. Well, those combinations come together, so you have I superscript A, you have I superscript O. This thing right here is called a genotype. That genotype is what you have in your body. It's this allele combination. It's a combination of genes that are going to determine your blood type. Well, without getting into too much detail, A is a dominant gene over the O. So this will result in you having a phenotype, the physical expression of blood type A. So that is in fact the phenotype. So let's recap real quick. Genes determine your traits. If there's different versions of a gene in a population, each one of those genes is given the term of an allele. When alleles are passed down from parents, meaning one from mom, one from dad, they come together and they form a genotype in that new offspring. That genotype right here is the determining factor in what type of physical expression you will have, or the phenotype. So talking about that, what we end up with is different genotype combinations that result in categories of phenotypes. So like I said here are all the possible genotypes. So if you basically just considered one of these coming from mom and one of these coming from dad, it does not matter which one comes from which, because the allele combinations that come together to form the genotype have set rules. For example, A is a dominant allele. B is a dominant allele. And that's evident because right here and right here, we know that even though maybe mom passed down an A allele or a B allele, and dad passed down an O allele, the blood type that results is still blood type A, or in this situation, blood type B. It does not matter because A or B will dominate over the O allele. Now, there's a tricky combination here that is involved with codominance. Codominance means if the right allele combinations come together, you actually don't end up with one dominating the other. Instead, they cooperate together, and that's when you end up with a combination of the two. So, for example, in that situation, our codominant one would be, boom, blood type AB, because if a mom or a dad gives the A allele and a mom or a dad gives the B allele, you end up with a combination of the two, which is blood type AB. The last situation is since we have a dominant allele in A or B, and then we have codominance when they're together, in blood you actually end up with a recessive one also. O ends up being the recessive one. So the only way you can end up with blood type O is if a mom and a dad both pass down an O allele to their offspring, resulting in blood type O. Why is this important? Well, if you know the blood type of the parents, and you know the blood type of an offspring, you can actually determine the exact alleles that each parent carries and the possible allele that their offspring carries. But what does that mean for us here in the forensics crime lab, or just in biomedical science in general? Here's why this is important. Let's say you're in some freak chainsaw accident, like for example, you're trying to cut the root ball off of a giant tree while it's being held by a forklift and you are using a single chainsaw unintelligently. And well, let's just say you cut your entire arm off. Well, to keep you from dying and bleeding to death, they are going to continually give you blood. Well, based on your genotype, you can only have certain types of blood. So in that instance, let's say you go to the hospital and you find out that you are in fact blood type A. You cut your arm off. They can't just go, oh, give me some blood. We got to fix this person. So it's not like they go to the refrigerator and grab whatever is all available. And they're like, oh, man, we're fresh out of OJ, but we got, the purple drink. No. Here's the thing. you can only receive certain types of blood. If they give you the wrong blood, you dead. So people with blood type A can only receive A or O. Now this should seem not obvious, but a little bit easier to understand why you can receive O if you are blood type A. It's because A dominates over O. It also has to do with some other factors, antibodies and antigens and the reactions between those, but we're not getting into those in this particular level of biomedical science. But just know that the reason it's important to know these different characteristics and the genotypes and phenotypes of blood is, one, it helps us in crime scene identification, but two, it helps us in the medical field where if there is injury or blood transfusions need to happen, we actually have the ability to make intelligent selections to keep that person alive. So, what does it mean for the crime scene? Let's just think about that. So, let's say that when blood is collected at the crime scene, the blood type is actually used as a basic way to determine ownership of the blood. Does it determine the exact owner? Absolutely not. Does it guarantee ownership? Absolutely not. This is because there are only 8 total blood types for the entire human population. And unfortunately saying, with 7 billion plus people on this planet, there's a really solid chance that you're going to have the exact same blood type as somebody else on this planet. So, for example, just because you can determine blood type does not guarantee ownership. Here's an example. Let's say down here in the stick figure, Miss Han is the stick figure with a sword, and she decides she wants to murder Riffy with that sword, but in the process happens to cut her hand on the sword used to stab Riffy. Both Riffy and Han, we both bleed all over the floor. Both of us have blood type A. Could you tell me definitively whose blood is actually on the floor? That answer is no. The reason the answer is no is because they have the same blood type. What would you do to actually figure that out? DNA analysis. So, that's blood. Let's talk about shoe prints, something a little bit easier because there is no genetics behind shoe prints. Let's talk about shoe prints. Because footwear impressions are virtually found anywhere in a crime scene, and a good forensic team can actually lift what are called latent footprints off of the floor. If you walk around barefoot, you can obviously pull up footprints associated with the skin, but we're talking about shoe prints more than anything. So there's 2 kinds. There's an impression that's left that's usually done when the floor is, or the ground is wet, soggy, where you can actually leave a depression in the floor. But there's also prints, which is just basically just like a fingerprint. It's not an impression or a depression on the object. It's just somehow some of that shoe print has been transferred and left onto the floor. So it is an actual significant form of physical evidence. So impressions left at the crime scene can provide a couple different things. 1, it can tell you where the crime occurred. 2, it can tell you the direction the suspect was traveling while entering, leaving, and in the event of like murder or something like that, or even robbery, you can actually determine where the crime was committed. It also helps place suspects at the crime scene and eliminate other suspects. When we're talking about the Ana Garcia case, this is one of the easier ones to do simply because you match it up based on tread pattern. So you looked at the tread pattern on the print, you looked at the bottom of the shoes, and boom, you figured out, hey, these are Ana Garcia's shoes. But here's the problem. There's a solid chance that somebody else on this planet bought those exact same shoes in the same area as Ana Garcia. Who's to say that somebody else didn't walk into her house and, completely kill her? Well, we can't say that for sure. So we go a little bit further. Okay, what kind of things can be pulled from a single shoe print or from multiple shoe prints in succession? You can get things like the wear pattern. Not everybody walks in the same way. Not everybody's heel to toe. Not everybody walks on the outside of their foot. Some people walk on the inside of their foot. Some people walk on the balls of their feet more. Some people are really heel to toe in terms of walking. So that wear pattern, the way the shoe wears out as the person wears it, is huge. There's the tread pattern. That's simply the manufacturer making treads on the bottom of the shoe. They come in patterns, they come in shapes. Nike's a big one. Every time you walk, you basically leave the Nike emblem somewhere. You have the size of the shoe, the width of the shoe. You have the make, the brand, the model, the mold, how a person distributes their weight while they walk. If they were running, if they were walking, you can determine stride length and say, hey, this person was running out of this crime scene. You can also determine the person's height based on stride length if it's determined that they were actually walking. In some instances, you can determine the exact location in which that shoe was purchased solely because there are things stuck in the tread. You can determine where they last were because of things stuck in the tread. Things that were stuck in the tread when they're in the crime scene can be left at the crime scene. So there's tons of things that can go down involving this. Here's what I found that was really cool in the FBI's website. The FBI's website brought this up. These are Air Force Ones. And they're basically saying, take a look at these 3 shoes. And if I said, which one of these shoes is different? Or are all these shoes the same? From looking at this picture right here, you'd kind of be like, pretty much they look the same. There's a couple weird differences, like you got this little spot right here, you got this wear spot right here. But here's the crazy part about this. If you actually zoom in on them and look, you'll notice that there are distinct differences in the mold in which these shoe treads were made. So you have the distance between the outside part of the tread and this inner lining or track right here. You'll notice that when you compare them, there's a wear right here, but also this inner line of the track disappears on the outside, where here is very definitive. And here you have a difference in the width of this track here compared to the width of the other 2. So by actually zooming in and look, you can see distinct differences that can give clues as to whose shoe it was. Maybe it's a gang, maybe they all wear Air Force Ones, but maybe one person got theirs from Foot Locker, the other person ordered theirs from Amazon. What happens is shoes are made in different areas, shipped to different areas, but those things can be traced back to where the factories were, the molds that you were used to make them. You have small differences in the shoe, boom, we got it. Let's talk about hair. Hair is, at one point, was a living tissue in the body, but then hair as it exists outside the body is dead. So hair is made from a couple other types of cells, but it's basically hardened by a protein called keratin. Keratin exists in hair, fingernails, and skin. It makes it hard and waterproof. But, unfortunately, in order for keratin to exist in those things, they are dead cells. Each species of animal that possesses hair has very characteristic lengths, color, root, shape of the hair, root appearance, and actual internal microscopic features that distinguish it from one animal to another. If you ever seen the movie Murder by Numbers with Sandra Bullock, they talk about hair fibers and they take them from a crime scene, and they actually find that some of the hair fibers at the crime scene were a baboon. Some of them were human. Well, the baboon hair fibers actually still end up being valuable, because who owns a baboon? Not many people. So by leaving baboon hairs at a crime scene, and as a baboon owner, you've really incriminated yourself in that particular case. But it also allows us to distinguish one animal from another, and it can actually use to distinguish from one individual another. For example, I might have dark hair, and then another classmate might have dark hair, but we are of different ethnic backgrounds. Believe it or not, those dark hairs actually can have definitive characteristics based on ethnic backgrounds. So hair evidence is collected with a couple goals in mind. One, identification. It tries to limit the persons of interest, but rarely does it ever identify the exact person of interest. There's only one case that hair can actually be used to identify the individual person themselves. The second thing is you want to compare the hair that you've collected to actual known samples. If you have a section or giant database of known samples of hair that you can compare one that you found, you can find out the animal, whether or not that person's male or female. You can find out ethnic background. You can find out their origin on this planet to a certain extent. So there's lots of things that it can do to help identify a substance, but it doesn't necessarily identify that suspect themselves. What is a hair made of? Essentially, you can break a hair down into 3 parts. You have the cuticle, the cortex, and the medulla. The cuticle varies in thickness. It can be thicker, it can be thinner. It depends on lots of things: male, female, hormonal balances, whether or not you're bald or not, things that are really awesome like that. But what happens when you look at it is that cuticle is that barrier to the inside portion of the hair, and it's kind of the protector, so it actually looks kind of scaly. So when you come into class and you want to look at your hair under the microscope, if you look at it, it's going to look kind of disturbing. And it actually varies in whether or not it has a pigment or color to it. So sometimes it's very translucent and you can pretty much see through it. The second layer is called the cortex. And remember, hair is 3-dimensional. So we're talking about working from the outside of the object in the round in. So the cortex is the next layer in. It also varies in organization. It has different densities, different sizes, and actually has pigment granules in there, and their distribution varies based on all of these different characteristics of male, female, ethnic background, all of that. One of the bigger things that they use for evidence in this particular case with hair is going to be the medulla. The medulla may be continuous throughout the entire hair fiber. It might actually start one place, stop in another, and then pick up again. It can be discontinuous, which is what I was just talking about. It can be fragmented, broken into a whole bunch of different sections, or it might not be there at all. You can't see it based on the actual color of the hair. A lot of people with blonde hair, the medulla is very hard to see. So if you compare the structure of a hair and you're trying to remember all these different layers, think about it like a pencil. So the inside lead portion of the pencil is the medulla. The cortex would be the wood portion of the pencil, and the cuticle, that outside covering, is the paint basically there. So, and that actually can help you remember which one's thicker or thinner and used for identification purposes. Everybody knows that one brand of pencil that you cannot use because the lead is terrible. You can identify that pencil solely based on the lead. Same thing with the medulla. Here's another cool thing that I found on the FBI's website that they can do with hair. Let's say that you found 3 different hair samples on a victim, and you want to know, gosh, these look completely different. Well, here's the thing. The only picture that you can actually use here to determine if, or actually determine down to the exact person that that hair belongs to, is going to be picture 3 right here. The reason being is if you look around this hair was forcibly removed from the person hard enough that they actually took some of the actual tissue from the scalp, arm, leg, wherever this hair came from. That is skin tissue. Keep in mind I said hair is dead. So all of this, you can't get DNA information from any of this because there is no viable DNA in there because the cells are dead. However, if a hair is ripped out forcibly with tissue around the root, you can actually take this tissue around the root, use it for DNA analysis, and boom, dwindle it down to an individual suspect possibly. This hair right here looks completely different from this one. However, the 2 here have slight differences in that this one has tissue. This hair here was naturally shed, meaning it just naturally fell out. This hair here was forcibly removed. We know that because there is actual tissue, skin tissue, around the hair follicle or hair root. This hair here was forcibly removed, however, it was broken and damaged the root. If you notice, there's only half of it there. These 2 here, forcibly removed, usually can indicate some kind of struggle. This one here, simply shed, so my chances of being incriminated in a crime scene are a lot less than some of the other people in class because I have no hair. But if I did, hairs fall out in a struggle just naturally, land on a dead body, boom, you've just incriminated yourself. Let's talk about fingerprints. Sorry this video is long, but it is what it is. Fingerprints are usually determined in the womb by 2 conditions. One, genetics, and the other, the conditions actually within the womb at the age in uterus of 10 to 15 weeks. It's not super old. You can't even determine gender typically by 15 weeks. It's usually around the 18th week that you can figure out the gender of an individual. So you have fingerprints before you know if they're male or female. No one person on this entire planet has the same fingerprint as another, because of the 2 factors we just talked about. But what we do find is there are ridge patterns that are consistent among the human population, and those ridge patterns really do have a pattern of inheritance between parents and children and offspring throughout family lines. Fingerprints themselves have 2 components. You have the raised ridges that actually leave themselves on countertops, glass, or things like that. And the reason you can see the raised ridges is because between them are furrows. Those are the low lines or valleys between all those ridges. Here's a nice close-up picture, and it even has sweat glands in there, which is kind of disturbing. But these right here are your ridges. These down here are your furrows. Let's talk about the 3 classification of ridge patterns. You have arch, loop, whorl. I need you to understand that the pictures in this slide are of fingerprints from a right hand. So here's your first one. You have a plain arch. What this is you end up with the ridges start on the left side or the right side of the thumb, finger, or wherever. They move directly across the pad of the finger, and right around the middle they just kind of go up a nice little hill, back down, and continue to complete opposite side of the finger. They don't form circles, they don't form loop-de-loops or anything like that. All you see is from left to right, a single lines forming a small hill in the middle and across. Then you have what are called a tented arch. Tented arches form more of a spike, or it looks like a tent that maybe you just got the first pole in the middle of the tent and it's sticking straight up. But a tented arch is the exact same as the above. You have it from the left side to the right side, but the spike in the center is significantly higher. So if we're looking at these 2 fingerprints right here, and I wanted you to tell me which one is a plane arch and which one is a tented arch. You're first looking for the lines that extend from one side, up a small hill, and down completely to the other side. But they both have them. However, the difference is in the size of that hill in the middle. This one here is going to be a plane arch. Small, nice rolling hill. This one here has a nice spike. You can even see that spike right here in this area. This one doesn't come to a spike. It's just got a nice easy hill. That's easy to understand, easy to identify. There's no loops, whirls, swirls, or anything like that. It's just a simple hill or a sharp, steep hill. Then what you have are whirls. Whirls, I think, are the easiest ones to identify. There's actually more categories of whirls. We're not going to get into that. But what they form is ridges. They form a circular pattern or a spiral pattern. I like to think that they look like dartboards more than anything. So if you look at this particular one right here, you get these loop, de-loop, de-loops, de-loop, de-loop. And you can also identify that by the fact that this little center section of the whirl doesn't actually point in a left or right direction, which would possibly indicate that it could be, instead of a whirl, it could be a loop. Let's get into the loops. You have two kinds, radial loop and ulnar loop. And it has to do with the direction the loop points. If it points towards the radius bone of your forearm, which is the one that connects to your thumb, if you hold your arms flat like this, the one right here is the one that connects to your thumb. The one on the outside is the one that connects to your pinky. Radius, ulna. That's why it's called a radial and ulnar loop. But they begin on one side of the finger, and they actually curve around, upward, and then down sloping to form towards the actual opposite end. In a radial loop, the loop itself will point in the same side of the hand as the thumb, towards the radius. The ulnar loop is the opposite. Does the same thing, curves, forms that loop, however, the loop slopes towards the pinky finger rather than towards the thumb. And the pinky finger is directly next to the ulna. Looking at these two pictures, if this is a right hand fingerprint, what you're looking at here is this one will point towards the thumb direction, making this one a radial loop. This one points towards the pinky direction, making this one an ulnar loop. In order to determine these things, it's really important that you actually know which hand the fingerprint came from. What makes fingerprints unique? You have the arrangement, you have the shape, you have the size, the number of lines, and then you have this very fancy thing called minutia points. These are places where the ridges change actual structure. They start, they stop, they branch off, they converge, they do all of these different things. In the LMS and in our instructions, we gave you 10 of the most common ones that you are going to find. There are several more. Of the 10 that we gave you, just so you know, a triple fork is the least likely to occur. Excuse me. While an ending ridge ending ridge is the most likely to occur. How do they use fingerprints? They use the minutia. Typically, they will identify 12 to 15 minutia in specific locations. They will pinpoint those in a computer search program, and they will run those minutia locations against a known database where other minutia have been identified. If we can get about 12 points of minutia common between two fingerprints, that's usually considered a pretty dang strong identification of the owner of that fingerprints. I like this little thing. Boom. We're done. I just finished. I hope you enjoyed it. If you didn't, I don't really care. It's helping the majority. If you have any questions, let me know.